transmitting 3, 4, 5, 6, and out of this 5 is V1 division of, that means ophthalmic division of trigeminal passes through it, and this cranial nerve along with this, which is passing through third cranial nerve, fourth, fifth V1 and sixth cranial nerve traveling through this SOF. So you have to remember and identify this foramen and which passes through it. Posterior to it, we have this space, we call them as sella turcica, housed by pituitary gland. So pituitary gland present in this particular region, we call them as sella turcica. Then in lateral to it, we find an foramen, which is round foramen. We call them as foramen rotundum. It's around by the rotundum, transmitting to V2. That means maxillary division of trigeminal nerve. Okay. Then little bit backwards to it, we find an oval opening, and next to that we find a tiny opening. So this oval opening, we call them as foramen ovale. Foramen ovale transmitting this cranial nerve number 3. Along with that, we have some kind of blood vessels runs through it. We call them as, in short, MALE. Middle meningeal artery, A for accessory, like accessory, what is that? L for lesser petrosal, E for emissary. MA. So maxillary, A for accessory meningeal. Okay. So MALE. So maxillary, accessory meningeal, L for lesser petrosal nerve, and E for emissary vein. So out of these 4 structure runs through this particular foramen ovale. The V3 division, which is important to us, which is the mandibular division that runs through it. So mandibular division, accessory meningeal artery, and this particular like lesser petrosal nerve and emissary vein runs through the cell. Then we have another opening medial to that. We call this medial opening as foramen lacerum. Lacerum do not transmit anything, which is passed by some cartilage structure. Under to it, we have some structure. We'll talk about that later. So if you move backwards to this particular foramen ovale, we have this foramen spinosum. So through the foramen spinosum, we have the meningeal artery which runs through it. We have seen the meningeal artery. When you cut the skull, we seen this meningeal artery, middle meningeal artery, we seen that. So this middle meningeal artery runs into this region with the dura mater, runs through it, anterior division, posterior division, runs through these particular divisions. Then we have this internal acoustic meatus. Through the internal acoustic meatus, runs through this cranial nerve number 7 and number 8, which is, we have seen that hanging down 7 and 8 together there. And then we have this 9, 10 and 11 cranial nerve comes to this particular foramen. We call them as jugular foramen. So this jugular foramen present in this region, and after that we have this region, we have foramen magnum, and inner side of it we have this 12th cranial nerve or hypoglossal canal present to it. So whenever you are seeing this particular skull, we have to identify this particular foramens in this particular specimen as well. So in the midline, we start with this space, we call them as cribriform plates, cranial nerve number 1. Then we have this optic canal, optic nerve along with the covering. Then lateral to it, we have SOF, which transmitting, what do you know? Posterior to that, we have rotundum. Rotundum is transmitting the cranial nerve V2. Then we have this one, ovale, oval structure. Foramen ovale is male structure like mandibular division, accessory meningeal artery and emissary vein and lesser petrosal nerve, which is transmitting through it. Posterior to it, we have an opening, we call them as spinosum. Medial to it, we have the lacerum. Lacerum fortunately do not transmit anything. Foramen spinosum transmit middle meningeal artery. And if the accessory meningeal not going through the male, it may transfer through this spinosum. Then we have this posterior to it, we have this internal acoustic meatus, we have this cranial nerve 7 and 8 coming through this internal acoustic meatus. Then we have this foramen, jugular foramen, which is present in this region, posterior to it, which is transmitting 9, 10 and 11, and hypoglossal canal on the inner side. We have it, which is transmitting the cranial nerve number 12. And then we have the foramen magnum, the largest opening. Through the largest opening, foramen magnum, transmitting through it is the medulla oblongata with the covering, and we have the blood vessels runs through it. We call them as vertebral artery, right and left uniting together forms the basilar artery, and as well as this accessory spinal nerve, which is spinal accessory, climbs up through it. So these are the major contributory structure that is coming through it. So whenever you are seeing this front to back, you have to identify this anterior cranial fossa, middle cranial fossa is housed by so many number of these particular openings. Out of this particular opening, individually you should be able to identify in the specimen given, and you should be able to identify what structure runs through it. Posterior cranial fossa is lined by this internal acoustic meatus and as well as jugular foramen. These are the two things that commonly been tested in examination, and hypoglossal canal, 12th cranial nerve, which is present in the inner side of this foramen, which call them as foramen magnum, and the foramen magnum transmitting the brainstem, which is known as the middle oblongata, which continues outside as a spinal cord, along with the covering of the meninges, along with the blood vessels which is going to supply the brain, along with this particular cranial spinal accessory part of the 11th cranial nerve comes out of it or climbs up out of it. So this is what is expected of you whenever you are identifying it. So if you flip it, if you see this particular thing from the base upside down, wait a minute, yeah, in this side. So if you flip it, then we find another foramen out here where I am putting the cursor. So you identify the styloid process and identify the mastoid process. In between them, we have a tiny slit. We call them a stylomastoid foramen. This tiny slit again high yield, because through it passes cranial number 7. Cranial number 7, which is going to supply all the muscle of facial expression. So it runs through this particular, this tiny foramen, tiny slit between them, we call them as stylomastoid foramen, and after coming out it enters to this particular parotid gland, splits into 5 divisions upside down, and then it supplies to the muscles of the facial expression, different muscles which is present in. So this particular opening that we have to identify whenever this wing is flipped inside out, or when this particular skull or base of the skull is flipped on it, and along with that we have to identify this particular foramen ovale, which can be seen, spinosum, which you can see that, and foramen lacerum is present in here, and jugular foramen is nearby in this region. So this midline swelling or midline opening as we are seeing here is foramen lacerum, into which we have this particular cartilage which is covering this particular region. We have a better picture, like, let's see. We do not have a better picture on it. Now see that we have an opening for the passage of the carotid artery. So we call this as a carotid canal. So this particular carotid canal is important for us. So this carotid canal, right side and left side, you should be able to identify beside this stylomastoid foramen. Stylomastoid foramen is a tiny slit which can be seen here as well. So this one with the pencil which is fitted in is a stylomastoid foramen, and this one where we have put the wire to it, we have this optic canal or opening of the optic canal which transmits this carotid canal which transmits this carotid artery or internal carotid artery. So these two openings are important, or rather very important. You should be able to identify that in the specimen whenever you are seeing it. हमारे sectional picture भी जो नहीं होता है. Alright, so this is zoomed out here. So foramen lacerum, don't get mixed up with the foramen lacerum with this CC, carotid canal, and this stylomastoid foramen is present in this particular region. So see this particular specimen, in it you should be able to identify this particular stylomastoid foramen. This will be given to you in examination, and carotid canal as well for identification. So this is about this particular openings which is present in this particular regions of the interior of this particular or base of the skull region. So these structures that you have to know, and along with that you have to remember the চলে যা আমারও লাগছে আমিও চলে যাব and cranial nerves in sequence, that what you have to keep in mind, like the names of it. So this particular foramens and the contents, they are what they are transmitting, that you have to understand, that you have to know, besides identifying them physically. So this is the first part and this is the second part. So this is started in any and every book. You should be able to get them and you should be able to identify them physically. So identify each of these foramen, make sure you mark it